It's an incredible paradox if you really stop to think about it. I want you, the listener, to just think about something as simple, as completely thoughtless as taking a step. You're just walking down the street, maybe checking your phone, totally unaware of your own body.
Right, yeah. You don't even register it.
Exactly. But that completely mundane action relies on this microscopic, insanely intricate chain of proteins, and they are bearing massive mechanical loads. And what's truly staggering, like what really keeps me up at night when I read about this, is that just one single misaligned genetic letter can ruin it all.
Oh, absolutely. One typo in millions.
Yeah. One microscopic typo in a sequence of literally millions of base pairs, and it brings that entire physical architecture crashing down. It can cause some of the most profound and devastating diseases known to medicine. So, welcome to this specialized deep dive on the Enkephalikos channel. I'm so glad you're here with us today to really get into this.
It really is a profound paradox. And, it's a real privilege to be here to unpack it with you today. Because when you start to look closely at the structural integrity of human movement, you realize it's an absolute marvel of biological engineering. And like any highly complex machine, there are very specific, incredibly fascinating ways that it can break down.
Exactly. And that mechanism of breakdown is exactly what we're dissecting today. So, for everyone listening, we're concluding our three-part exploration into Chapter 57 of the 6th edition of Principles of Neuroscience.
Right, the grand finale of the chapters.
Very grand finale. We've been looking deeply into diseases of the peripheral nerve and motor unit. Today, we're taking everything we've learned and zooming in on the final piece of the puzzle, which is the diseases of the skeletal muscle itself, both the inherited and the acquired forms.
And we'll be wrapping up with the major highlights of this entire field of study, right?
Yes, exactly. And my goal today is to really dig into the why and the how. Like, I don't just want to know what these diseases are called. I want to understand the biophysics. I want to understand how a single atomic change in an ion channel can leave someone completely paralyzed.
Well, that is exactly the level of detail we're going to get into. In our previous deep dives, we spent a lot of time on motor neuron diseases and peripheral neuropathies.
Right, the electrical cables, basically.
Exactly, the cables. But now we're moving our focus entirely away from the nervous system. We're looking squarely at the muscle fiber itself.
Okay, so let's establish our baseline here. If we're looking at a patient who is experiencing profound muscle weakness, how are we defining a myopathy in this context? Because I think the intuitive leap for you or me or anyone listening is that if you can't move your arm, your brain or your nerves must be failing.
Yeah, that's the natural assumption.
So how do we distinguish a true myopathy from the neurogenic diseases we talked about in parts 1 and 2?
It's the absolute foundational distinction in neuromuscular medicine. A myopathy is defined strictly as weakness caused by the degeneration or dysfunction of the muscle fibers themselves. That is the key.
Okay, so just the muscle.
Right. This is completely distinct from a neurogenic disease like ALS or a peripheral neuropathy. In those neurogenic conditions, the nerve fails and the muscle wastes away simply because it lacks stimulation.
So it's essentially dying of neglect.
Exactly, dying of neglect. But in a myopathy, the software and the wiring are flawless. The motor neuron in the spinal cord is perfectly healthy. It's firing the correct signal.
Wow.
The nerve axon conducting that signal down your arm or leg is perfectly intact. The neuromuscular junction is releasing its neurotransmitters normally. The failure occurs intrinsically within the hardware of the skeletal muscle tissue itself.
Okay, let's unpack this. The command is arriving loud and clear, but the muscle just physically cannot execute it. It's like the engine receives the spark, but the cylinders are broken. What does that progression actually look like at the cellular level? Like if I were to look at a muscle biopsy of a myopathy under a microscope, what is the story being told in that tissue?
It's a story of a microscopic battle of attrition. And initially, the body fights back really hard. When muscle fibers first begin to degenerate, whether that's from a genetic defect or an autoimmune attack, the body attempts to compensate.
Because muscle can regenerate, right?
It has a remarkable capacity for repair, yeah. The tissue contains satellite cells, which are basically muscle stem cells. They activate to replace the missing or damaged fibers. So if you do a biopsy in the early to middle stages of a myopathy, you'll actually see a very chaotic histological picture.
Chaotic how? Like lots of different things happening at once.
Exactly. You'll see signs of active degeneration, so fibers that are swollen, necrotic, and being eaten away by specialized immune cells called macrophages.
Okay, the cleanup crew.
Right. But simultaneously, you'll see active regeneration. You'll find New, small, immature muscle fibers trying to bridge the gaps.
Oh, wow. So it's like a city constantly trying to rebuild buildings while an earthquake is still actively happening?
That is a very apt way to describe it.
Hmm.
However, as the disease progresses relentlessly, this renewal process ultimately hits a wall. The stem cells just get exhausted, or the environment becomes too toxic.
So the regeneration simply can't keep pace with the rate of destruction.
Right.
And once that tipping point is reached, what happens to the muscle bulk? It doesn't just leave empty space behind, does it?
No, nature abhors a vacuum.
Huh.
As that renewal fails, the muscle fibers are progressively and permanently lost. But the volume of the muscle might not decrease immediately,
Why not?
because the lost contractile tissue is replaced by something else. Often you see a chronic inflammatory cellular response. And in many long-standing myopathies, the dead muscle tissue is infiltrated and replaced by fat cells and dense fibrotic connective tissue.
Ugh, scar tissue.
Yeah, so the muscle might look normal in size from the outside or even enlarged in some cases.
Mhm.
But internally, it's turned from a highly organized contractile machine into a chaotic mix of fat and scar tissue.
That is a stark visual for everyone listening. Now, let's talk about how you actually diagnose this in a living patient before you ever take a biopsy. We discussed electromyography, or EMG, extensively when we covered peripheral neuropathies.
Right, the electrical testing.
Yeah, where you insert that fine needle electrode directly into the muscle to listen to its electrical activity. If you stick that needle into the muscle of someone with a primary myopathy, how does that electrical signature differ from someone with a dying nerve?
What's fascinating here is how clearly the microscopic anatomy dictates the electrical signal you see on the screen. To understand the EMG, we have to quickly revisit the concept of the motor unit.
Okay, let's do.
So one single motor neuron coming from the spinal cord and all the individual muscle fibers it branches out to control.
Right, it's a team.
Mm,
One captain, many soldiers.
exactly. Now, in a neurogenic disease, the motor neuron, the captain, dies. So you lose entire motor units, entire teams are wiped out.
But the nervous system tries to fix that, right?
Yes, it's very clever. The surviving motor neurons nearby will sprout new axonal branches to essentially adopt the orphaned muscle fibers left behind by the dead neuron.
So the surviving captains take on extra soldiers to compensate.
Yes. So on an EMG of a neurogenic disease, you see fewer total motor units firing because neurons have died. But the ones that do fire produce massive, prolonged electrical signals. Neurologists call them giant units.
Because one nerve is driving way more muscle than it should.
Exactly. This happens because one single nerve is now depolarizing an abnormally large number of muscle fibers all at once. The signal is huge.
But in myopathy, you said the nerves are completely fine, so the situation is inverted.
Completely inverted. The captains are healthy, so the number of motor units under voluntary control is entirely normal. When the patient tries to flex the muscle, all the motor units fire. However, within each of those motor units, individual muscle fibers, the soldiers, have died off randomly.
Ah, I see. So the team is still there, but it's severely depleted. Instead of a captain commanding 100 soldiers, he's shouting orders to maybe 30.
Precisely. And because there are fewer functioning muscle fibers per unit, the compound motor unit potentials recorded on the EMG look completely different. Instead of those giant units, you see electrical spikes that are of very brief duration and much smaller amplitude.
Because there's just less physical muscle making electricity.
Exactly. There's simply less electricity being generated because there's less muscle mass responding to the nerve's command.
And the text also mentioned they look polyphasic. What does that mean in this context?
Think of a healthy motor unit, like a highly trained choir. When the conductor signals, they all sing the exact same note at the exact same millisecond.
Perfect synchrony.
Right. On an EMG, that synchrony creates a clean, sharp, single electrical peak. But in a myopathy, the surviving fibers within that motor unit are sick. Some might be regenerating and tiny, some might be scarred and sluggish. They don't depolarize at the exact same speed anymore.
So they're out of sync. The choir is singing the same song, but everyone is a fraction of a second off from each other.
Exactly. And electrically, that means the action potential isn't a single clean spike. The electrical wave on the monitor fluctuates up and down multiple times as the signal stutters across the varying health of the fibers.
Oh, so it has multiple peaks.
Yes, that multiple peaked wave is what we call a polyphasic potential.
Okay, so when you see brief duration, small amplitude, and a stuttering polyphasic wave, you know you aren't looking at a nerve problem. You are seeing the electrical signature of a physically depleted, struggling muscle.
You nailed it.
That makes perfect sense. Now, the text breaks these primary muscle diseases down into two broad categories, acquired and inherited. Let's start with acquired. What does it mean to acquire myopathy, and what's the textbook prototype for this?
An acquired myopathy means the patient was born with perfectly healthy genetics for their muscles, but some external or systemic factor has come along and damaged them.
Like an infection or something.
It could be a toxin, a severe metabolic disturbance, or, very commonly, an autoimmune attack. And the quintessential acquired myopathy discussed in the text is dermatomyositis.
Dermatomyositis. Okay, breaking down that word for you listening. Dermato means skin, and myositis means muscle inflammation. So this is a disease that hits both the skin and the muscles simultaneously.
Correct. It's defined by a very specific dual clinical presentation. The rash is highly characteristic. Patients often develop what is called a heliotrope rash.
What does that look like?
It's a purplish erythematous discoloration, primarily on the face, often right around the eyelids. They also frequently get scaling, red rashes, over the extensor surfaces of their joints, like the knuckles on their fingers, the elbows, and the knees. We call those Gottron's papules.
Gottron's papules, okay. And while this is happening on the outside, what is happening on the inside?
Simultaneously, or sometimes closely following the onset of the rash, the patient develops a profound myopathic weakness. And this weakness has a very specific pattern. It primarily affects the proximal limb muscles.
Meaning the muscles closest to the center of the body, like the trunk, the shoulders, the hips, and the thighs, not the hands or the feet.
Exactly. They might find they suddenly have trouble combing their hair because they can't keep their arms raised.
Right.
Or they struggle to stand up from a low chair without using their arms for leverage.
Because the hip muscles are failing.
Right. This weakness can progress quite rapidly over a matter of weeks or a few months, and it can range from mildly debilitating to life-threatening if it begins to affect the muscles of swallowing or respiration.
Okay, let's look at the underlying logic here. If this is a myopathy, the muscle fibers are degenerating. But why the skin rash? If my immune system decides to attack my muscles, why are my eyelids turning purple? What's the actual underlying mechanism connecting these two completely different tissues?
It's a brilliant question, and the answer lies in understanding what the immune system has actually chosen as its target. Dermatomyositis is indeed an autoimmune disorder, but here's the twist. The immune system is not attacking the muscle fibers directly.
Wait, really? But the muscles are what's dying.
They are, but they're collateral damage. The immune system in dermatomyositis is specifically targeting the endothelial cells that line the small blood vessels, the tiny capillaries and arterioles.
Oh.
Yeah, these are the blood vessels that weave through the connective tissue supplying both the skin and the intramuscular spaces. The immune system unleashes a cascade of antibodies and complement proteins that physically destroy these capillaries.
Ah, I see. This totally flips the perspective. If the muscle fibers are the engine of a car, dermatomyositis isn't the immune system taking a sledgehammer to the engine block itself. It's the immune system selectively attacking and dissolving the fuel lines, the blood vessels, that are feeding that engine.
That is a perfect analogy. The muscle damage in dermatomyositis is entirely secondary to ischemia.
Ischemia, meaning lack of blood flow.
Right. Ischemia means a critical lack of blood flow and consequently a lack of oxygen and nutrients. When the capillaries are destroyed, the muscle fibers are literally starved to death. They undergo ischemic necrosis, which we then clinically observe as that profound proximal weakness.
And because the same small blood vessels supply the skin.
Exactly. Their destruction causes the characteristic rashes.
That is deeply fascinating, that the immune system can essentially acquire a target that results in the starvation of the muscle rather than a direct assault on the muscle itself. It's a siege tactic. And I remember reading in the text that there's often a sinister trigger for this autoimmune confusion, especially in adults.
Yes, that's a very important clinical point. While dermatomyositis can affect children in a standalone juvenile form, in adult patients, about 10 to 15% of cases are actually hiding an underlying malignant tumor.
Wow.
Yeah, often an occult cancer of the lung, breast, or gut that hasn't even been diagnosed yet.
So the body detects a cancer, ramps up an immune response to fight the tumor, and somehow those anti-tumor antibodies accidentally cross-react with the blood vessels in the skin and muscle.
Precisely. It is what we call a paraneoplastic syndrome. The immune system's attempt to save the patient from cancer inadvertently causes a devastating autoimmune disease in the muscles. It highlights how exquisitely sensitive
Yeah.
and sometimes dangerously imprecise our immune surveillance can be.
It's incredible. But while the immune system can acquire a target and cause this ischemic destruction, the text makes it very clear that many of the most devastating and relentless myopathies are not acquired at all.
No, they're not.
They are coded into our DNA from the very moment of conception. The blueprint itself is flawed. And that brings us to the core of this chapter, the inherited muscular dystrophies.
Yes, moving from the acquired inflammatory myopathies to the genetic ones. The muscular dystrophies are a specific large class of inherited diseases. The defining hallmark of a dystrophy, as opposed to other genetic muscle diseases, is that the weakness is progressive and relentless.
It never stops.
Right. It's not just that the muscle is built poorly. It's that the muscle actively tears itself apart over time.
And when we talk about muscular dystrophies, there is one that casts a massive shadow over the entire field. It's the most well-known, the most extensively studied, and arguably the most severe, Duchenne muscular dystrophy, or DMD.
Duchenne really is the prototype for understanding inherited muscle disease. Clinically, it presents a very specific and tragic progression. First, we have to look at the inheritance pattern. It's an X-linked recessive trait.
Okay, let's clarify what that means for you listening. X-linked recessive means the genetic mutation is located on the X chromosome. Because biological females have two X chromosomes, if one carries the mutation, they have a backup copy on the other X chromosome.
Exactly. And that backup usually provides enough healthy protein to prevent the disease. They might be carriers, but they usually don't show severe symptoms.
But biological males only have one X chromosome and one Y chromosome. If their single X chromosome has the Duchenne mutation, they have no backup.
Right. They will get the disease. Because of that, Duchenne almost exclusively affects boys, occurring in about 1 in every 3,500 to 5,000 male births.
And the timeline is just heartbreaking.
It is. The clinical timeline is heartbreakingly predictable. The disease usually starts to show its first subtle symptoms in early childhood, around age 2 or 3. The parents might notice the toddler is delayed in walking, or they fall down frequently. They develop a characteristic waddling gait.
And there's a very specific physical maneuver they do to stand up, isn't there? The text mentioned Gower's sign.
Yes, Gower's sign is a classic clinical indicator. Because their proximal muscles, their thighs and hips are already weakening, a child with Duchenne cannot simply stand straight up from a seated position on the floor.
They don't have the strength.
No. Instead, they have to roll over onto their hands and knees, push their hips up in the air, and then literally walk their hands up their own legs to push their torso upright.
Oh, wow.
They're using their arm and back muscles to compensate for the failing hip extensors.
That is such a vivid and difficult image. And because this is a dystrophy, it doesn't stabilize. It just gets worse.
It's relentless. As the child grows, the muscle degeneration outpaces any growth or repair.
Yeah.
Typically, by the time these boys reach the age of 12, they have lost the ability to ambulate and require a wheelchair.
That's so young.
And tragically, the disease does not only affect the skeletal muscles of the arms and legs, it also eventually destroys the diaphragm, which controls breathing, and the cardiac muscle of the heart. Duchenne is usually fatal in the 3rd decade of life, typically due to respiratory or cardiac failure.
It's a devastating reality. But the text immediately contrasts Duchenne with another condition called Becker muscular dystrophy, or BMD. And this is where the biology gets incredibly fascinating, because Becker is intimately related to Duchenne, but the clinical picture is vastly different.
It is. Becker muscular dystrophy is also an X-linked recessive disorder, and crucially, it is caused by mutations in the exact same gene that causes Duchenne.
The exact same gene.
The exact same one. However, the phenotype, the actual physical manifestation and severity of the disease is much, much milder.
How much milder?
Significantly. The onset of weakness in Becker happens later in childhood or even into adolescence. While they do experience progressive weakness of those same proximal leg and arm muscles, it's a vastly slower progression. Individuals with Becker dystrophy are typically able to walk well into adulthood, often into their 40s or 50s.
Wow.
Yeah, and they have a significantly longer life expectancy than those with Duchenne.
Okay, so this is the central mystery we need to solve right now. If Duchenne and Becker are caused by mutations in the exact same gene, on the exact same chromosome, why is one a rapid childhood death sentence while the other allows a patient to walk for decades? To understand that, we need to look at the genetic culprit itself.
We do. The culprit is the DMD gene, located on a specific band of the X chromosome called XP21. And to truly grasp the difference between Duchenne and Becker, we first have to appreciate the sheer, incomprehensible scale of this gene.
How big are we talking?
The DAD gene is an absolute behemoth. It spans approximately 2.5 million base pairs of DNA.
2.5 million base pairs just for one single gene. That number is really hard to wrap your head around.
It's the second largest known gene in the entire human genome. To put it in perspective, this single gene accounts for about 0.1% of the entire X chromosome all by itself. It is massive.
Why is it so big? Is it just a massive string of instructions?
Actually, no. Like most human genes, the DMD gene is broken up into coding regions called exons and non-coding regions called introns. You can think of introns as spacer DNA.
Okay, spacer DNA.
When the cell wants to make the protein, it transcribes the entire massive 2.5 million base pair sequence into a precursor messenger RNA. But then it has to edit it. The cell's machinery cuts out all the non-coding introns and splices only the exons together to make the final, mature mRNA blueprint.
So the exons are the actual meaningful sentences, and the introns are just blank pages between them.
Exactly. And the DMD gene has at least 79 exons spread across those 2.5 million base pairs. Now, because this gene spans such an enormous physical distance on the chromosome, it is a massive target for spontaneous mutations during cell division.
It's simply a statistical reality. The bigger the target, the more likely it is to take a hit.
Right.
And what kind of hits are we talking about here?
The most common mutations causing these dystrophies are deletions. Large chunks of the DNA, often spanning one or several entire exons, are simply missing. They get deleted by accident when the DNA is being copied.
Okay, so we have this gigantic 79 exon gene, and occasionally a chunk of it gets deleted. How does a deletion cause Duchenne in one patient and Becker in another?
This is where we get into the elegant mathematical precision of molecular biology. It all comes down to a concept called the reading frame.
The reading frame. Okay.
When the mature mRNA goes to the ribosome to be translated into a protein, the ribosome reads the genetic code in sets of 3 nucleotide letters at a time. These 3-letter sets are called codons. Each triplet codon codes for one specific amino acid, which are the building blocks of the protein.
Right. I always visualize this as reading a sentence where every single word is exactly 3 letters long. Like, the cat ate the rat and the bat.
That is the perfect way to visualize it. The ribosome moves along, reading 3 letters, adding an amino acid, moving to the next 3 letters, adding another.
Okay, so it just marches down the line.
Right. Now imagine a deletion occurs in the DMD gene. In Duchenne muscular dystrophy, the deletion typically disrupts that reading frame. We call this a frame shift mutation.
A frame shift? How does that actually happen?
Let's say a chunk of DNA is deleted, and the number of base pairs lost is not perfectly divisible by 3. For example, let's say exactly 4 base pairs are lost. Or maybe one entire exon that happens to contain a number of letters not divisible by 3 is deleted. When the cell splices the remaining exons together to close the gap, the sequence is ruined. The 3-letter words downstream of the deletion are no longer aligned properly.
Ah, I see. Let's go back to my sentence. The cat ate the rat. If I delete just one letter, say the C in sati, the ribosome doesn't know there's a space missing.
No, it just keeps reading in blocks of 3.
So the cat ate becomes the eta tet her rat.
Exactly. From the point of the deletion onward, the code is complete and utter gibberish. Because the reading frame is shifted, the ribosome starts inserting completely incorrect amino acids into the protein chain.
It's building nonsense.
Yes, nonsense. And very quickly, this randomized shifted frame will accidentally generate a stop codon, a specific 3-letter sequence that tells the ribosome, halt translation immediately, the protein is finished.
Even though it's nowhere near the actual end of the gene, so the cell produces this severely truncated, stubby little protein that is mostly made of the wrong amino acids anyway.
Yes. And the cell's internal quality control mechanisms are very strict. They recognize this truncated, misfolded protein as defective and immediately degrade it. The end result of a frameshift mutation in Duchenne is that functional dystrophin protein is almost entirely absent from the muscle cell. You have 0 functional protein.
But Becker is different. Becker preserves the reading frame.
Correct. In Becker muscular dystrophy, the deletion mutation, even if it is a massive deletion that removes several entire exons, happens to remove a number of base pairs that is perfectly divisible by 3.
Oh, so if you delete exactly 3 letters, or 6, or 90, you don't mess up the alignment of the downstream text.
Exactly. Therefore, when the slanking exons on either side of the deletion are spliced back together, the reading frame remains perfectly intact.
Okay, I want to try an analogy here to make sure this crystallizes for everyone listening. Let's imagine the DMD gene is a massive, highly detailed instruction manual for building a machine.
Okay, I like that.
A Duchenne frameshift mutation is like tearing a page in half right down the middle of a sentence and then gluing the book back together. Because you've shifted all the text over by half a line, every single page after the tear becomes unreadable gibberish. The instructions are ruined, you can't build the machine, you have to throw the whole book out.
That's right.
But a Becker in-frame mutation is like carefully ripping out an entire chapter, say, chapter 5, and taping chapter 4 directly to chapter 6. You completely lose whatever specific instructions were in chapter 5, but when you flip the page from 4 to 6, the grammar still makes sense. The sentences still read correctly. You can still read the rest of the manual and build most of the machine.
That is a brilliant and highly accurate way to conceptualize it.
Yeah.
In Becker, the cell reads the mRNA and produces a dystrophin molecule that has an internal deletion. It's literally missing a chunk out of its middle section.
Right.
However, because the beginning and the end of the protein are still correctly translated and structurally sound, this shortened dystrophin is allowed to persist by the cell.
It's a smaller, stubbier version of the machine, but it still basically works.
Precisely. It retains partial function. It's not perfect, which is why patients with Becker still experience progressive muscle weakness. But having a partially functional, internally deleted protein is vastly superior to having no protein at all. It is enough to significantly ameliorate the mechanical stress on the muscle and drastically slow the degeneration compared to Duchenne.
That structural insight that it all comes down to the math of being divisible by 3 is incredible. And what's even more thrilling is that this exact understanding of the reading frame isn't just trivia, it's actively paving the way for some of the most cutting-edge science fiction level therapies in neurology today, right?
It absolutely is. For decades, Duchenne was considered an untreatable death sentence because, how do you fix a missing gene? Especially when the DMD gene is 2.5 million base pairs long.
Yeah, you can't exactly just mail a new copy in.
No, you can't simply pack the full healthy DMD gene into a standard viral vector like an adeno-associated virus, or AAV, which we often use for gene therapy. The gene simply will not fit. The virus's cargo capacity is much, much too small.
It's like trying to fit an entire encyclopedia set onto a floppy disk. It won't work. So what are the workarounds? The text mentions a few incredibly clever therapeutic frontiers.
One of the most successful approaches relies directly on exploiting that difference between Duchenne and Becker that we just discussed. It utilizes synthetic molecules called antisense oligonucleotides, or AS.
ASOs, okay.
The overarching goal of ASO therapy in this context is to induce a phenomenon called exon skipping.
Exon skipping. Okay, let's break down how that actually works in a patient.
Let's use a very common real-world example. Let's say a young boy has Duchenne because he has a deletion of exon 50. Now, because exon 50 is missing, when the cell tries to splice exon 49 to exon 51, the reading frame is shifted.
Right, the grammar is broken.
This specific frame shift creates a premature stop codon right inside exon 51, halting production and causing severe Duchenne.
Okay, so the deletion of 50 ruined the grammar for 51.
Exactly. The idea behind the ASO therapy is to administer a custom-designed synthetic piece of nucleic acid, the ASO, that is engineered to bind specifically to the patient's pre-mRNA transcript, right at the splice site for exon 51.
So you inject this synthetic molecule, it travels into the muscle cell nucleus, finds the RNA copy of the DMD gene, and literally sticks to exon 51 like Velcro.
Yes. And by binding there, the ASO acts as a physical roadblock. It effectively hides exon 51 from the cell's molecular splicing machinery. The spliceome comes along, sees exon 49, doesn't recognize exon 51 because it's covered up, and so it just skips it entirely and splices exon 49 directly to exon 52.
Wait, so the cell just completely leaves out exon 51?
Yes.
But doesn't that make the protein even shorter? Why would you want to delete more of the gene?
Because of the math of the reading frame. While the deletion of exon 50 alone causes a frame shift, researchers calculated that if you artificially force the cell to also skip exon 51, the combined deletion of both 50 and 51 happens to be a multiple of 3 base pairs.
Oh, wow. So you are intentionally introducing more genetic deletion at the RNA level to fix the grammar of the sentence.
Exactly.
By skipping 51, you restore the reading frame for exon 52 and the rest of the gene downstream.
Precisely. You are deliberately converting a severe out-of-frame Duchenne mutation into a milder, in-frame Becker-like mutation. The resulting protein produced by the boy's cells is now missing exons 50 and 51, so it is shorter than normal, but the reading frame is restored.
So it's readable again.
Right. The cell produces this partially functional dystrophin, which can dramatically slow the progression of the muscle wasting.
That is just brilliant, elegant bioengineering. You're hacking the cell's own editing software on the fly. Now, what about those viral vectors? You said the whole encyclopedia won't fit in the AAV virus. Have researchers found a way to deliver actual DNA instructions?
They have, by being ruthlessly efficient. Since the whole gene won't fit, the alternative approach is to deliver what we call mini-dystrophins, or microdystrophins.
Microdystrophins, how does that work?
Well, researchers look closely at the structure of the dystrophin protein in patients with very mild Becker muscular dystrophy people who are missing huge chunks of the protein, but were still walking in their 60s.
They looked at what was left.
Right. They realized that large sections of the central rod domain of the protein are actually somewhat dispensable for basic structural function. The truly crucial parts are the two ends: the amino terminal that binds to the inside of the cell and the carboxy terminal that binds to the cell membrane.
So they basically designed a stripped-down, highly condensed version of the gene.
Exactly. They engineered synthetic microgenes that only contain the crucial ends and a small portion of the middle. These microgenes are roughly 30% the size of the full gene, which is just small enough to be packaged into an adeno-associated virus.
So you infuse these engineered viruses into the patient. The virus infects the muscle cells, because viruses are naturally great at breaking into cells. But instead of delivering viral instructions to replicate, it unloads this microgene blueprint.
Yes. The microgene sits in the nucleus, and the muscle cell begins reading it, producing massive amounts of this highly truncated, yet highly functional microdystrophin protein. And that stabilizes the muscle fibers and halts the degeneration. It is a monumental achievement in gene therapy.
Okay, so we have talked extensively about the gene, the reading frame, and how we might hack it. But let's take a step back. What does this all mean for the actual cell?
Right, physically.
Yeah. We keep talking about this giant protein as a structural component, but what exactly does it do? Why does missing it cause the muscle to literally tear itself apart during normal movement? This transitions us logically into understanding the broader architecture of the muscle cell, because dystrophin doesn't act alone. Let's look at the dystrophin-associated complex.
To understand what dystrophin does, you have to look at where it lives and what it connects to. Dystrophin is localized entirely to the inner surface of the muscle cell's plasma membrane, which we call the sarcolemma. Its primary job is structural. I like to think of it as a massive, flexible shock absorber and a critical physical anchor.
An anchor connecting what to what? Paint the picture for me.
Okay, picture the inside of a muscle cell. It's packed full of myofibrils, these long, sliding filaments of actin and myosin that physically contract to generate force. The amino terminal end of the long dystrophin protein reaches deep into the cell and binds tightly to the cytoskeletal F-actin.
Which is the internal framework holding all those contractile proteins together.
Right.
So it's grabbing onto the internal moving parts.
Yes. Now, the other end of the dystrophin protein, the carboxy terminal, reaches up to the cell membrane. But it doesn't just stick to the fat of the membrane. It links firmly to a large, intricate cluster of other proteins that are embedded directly through the membrane, known as the dystrophin-associated glycoprotein complex, or DGC.
So dystrophin is a bridge. It connects the internal scaffolding of the muscle to the outer wall of the cell.
It's a bridge, but the network doesn't stop at the cell membrane. That transmembrane complex, which is made of multiple subunits with names like sarcoglycans and dystroglycans, reaches through the cell membrane and binds tightly to structural proteins in the extracellular matrix outside the cell.
Oh, it goes all the way out.
All the way. Primarily, it binds to a protein called laminin, which in turn binds to the surrounding collagen framework.
Okay, I see the full picture now. Dystrophin isn't just a beam floating in the cell. It's the linchpin of an immense structural scaffolding that spans from the deep, contracting interior of the cell, passes completely through the delicate membrane, and anchors securely into the rigid extracellular matrix on the outside.
Precisely, and this entire physically linked network is absolutely required to confer tensile strength to the muscle fiber.
Yeah.
Think about the physics of movement. When a muscle contracts, the actin and myosin slide together, generating massive mechanical force. If those internal contracting filaments are not securely anchored to the extracellular matrix on the outside, that force of contraction will simply pull against the fragile lipid membrane of the cell and literally rip it apart.
So dystrophin acts as the shock absorber, distributing that mechanical stress safely across the membrane to the outside matrix. So when dystrophin is missing, as in Duchenne, the membrane is incredibly fragile.
Yes. Every single time the boy flexes a muscle, every time he takes a step or even just breathes, the internal contraction inflicts microscopic tears in his own muscle cell membranes.
Wow.
And when the membrane tears, the tightly controlled internal environment of the cell is ruined. Massive amounts of calcium flood in from the outside, which activates destructive enzymes called proteases that literally digest the cell from the inside out, leading to the rapid necrosis we see clinically.
That is a staggering realization. The very act of moving is what destroys the muscle. And what's crucial to understand here is that dystrophin is just one protein in this chain. What happens if dystrophin is fine, but one of the other proteins in this massive complex is genetically mutated?
You've hit on a critical principle of this complex. A chain is only as strong as its weakest link. If a patient inherits a genetic mutation that destroys any of the proteins in this physically linked network, if they have a mutation in the alpha-sarcoglycan gene or the dystroglycans or even the laminin in the extracellular matrix, you destabilize the entire scaffolding.
So even if you have perfect dystrophin, if it has nothing to anchor to, it's useless.
Exactly. Disruption of this linked network due to a mutation in just one of the supporting proteins often leads to a secondary reduction in the levels of the entire complex at the membrane. The whole structure falls apart.
Ah, so they all depend on each other.
Right. This is why mutations in dozens of completely different genes can result in muscular dystrophies that look clinically very similar to each other. They're all breaking different links in the exact same mechanical chain.
Now, in exploring how these links break, the text brings in a really surprising organelle, the Golgi apparatus. When I learned basic cell biology, I was taught the Golgi is the cell's shipping and receiving center. It packages things up. How does a defect in a shipping center cause a severe muscular dystrophy?
It's a wonderful example of how interconnected cellular biology is. Those transmembrane proteins we just talked about, specifically alpha-dystroglycan, the one that reaches outside the cell, they don't just bind to the extracellular laminin matrix automatically. They need to be chemically modified first to make them sticky.
Sticky how?
They require the addition of very specific, highly complex sugar chains to their surface in order to chemically adhere to the laminin.
And adding those sugar chains, a process called glycosylation, is the primary job of the Golgi apparatus.
Correct. The Golgi houses specific resident enzymes whose only job is to build these intricate branching sugar trees onto proteins before they are sent to the cell membrane. The text mentions several specific Golgi enzymes like fukutin and POMT1. If a patient inherits a genetic defect in the genes coding for these Golgi enzymes, the muscle cell still perfectly synthesizes the alpha-dystroglycan protein backbone.
The protein is built fine, but the Golgi fails to sugarcoat it properly.
Exactly. It fails to glycosylate it. So the naked protein makes it to the cell membrane, but it is missing the chemical Velcro needed to stick to the extracellular matrix.
The mechanical link is broken. Even though all the structural proteins are present, they can't hold hands.
Yes, and the mechanical stress tears the cell apart, just like in Duchenne, leading to severe muscular dystrophy. And here is a fascinating and tragic clinical correlate.
What's that?
Because these exact same glycosylation enzymes in the Golgi are also absolutely critical for the migration of neurons during early fetal brain development. Genetic mutations in these Golgi enzymes don't just cause muscle weakness. They often cause aberrant brain development,
Oh, wow.
leading to profound cognitive impairment, seizures, and severe structural brain malformations alongside the muscular dystrophy. It's a systemic failure of protein modification.
It is truly amazing how a defect in attaching a microscopic sugar molecule can cause a system-wide collapse of both muscle and brain.
It really is.
Now, if dystrophin and its complex are the structural scaffolding preventing damage, the text also introduces a completely separate set of proteins involved in repairing damage. Because, let's face it, even healthy muscle gets damaged.
Absolutely. Normal, healthy skeletal muscle experiences microscopic membrane tears all the time, especially during eccentric contractions or strenuous exercise. Think about the soreness you feel after lifting heavy weights. Part of that is microscopic mechanical damage.
Right.
To survive a lifetime of use, the muscle cell must have a highly efficient, rapid response emergency repair mechanism. This is where proteins like dysferlin and its binding partner caveolin-3 come into play.
So if dystrophin is the structural steel beam holding the building together, dysferlin is the emergency repair crew waiting in the basement.
That's a great way to put it. When a breach occurs in the sarcolemma and calcium starts to leak in, that calcium spike acts as an alarm bell. Dysferlin is a protein central to generating and trafficking rafts of tiny intracellular vesicles.
Vesicles, like little bubbles of membrane.
Right. When the alarm sounds, dysferlin rapidly recruits these vesicles to the exact site of the injury. The vesicles coalesce, pool together, and fuse with the torn plasma membrane to rapidly patch the hole.
It's essentially throwing a biochemical tarp over the tear and sealing it before too much calcium can leak in and trigger the self-destruct sequence.
Precisely. It seals the breach in seconds. But what happens if you have a genetic mutation in the dysferlin gene?
Well, presumably you get another type of muscular dystrophy, but the mechanism of failure is entirely different.
Correct. In a dysferlinopathy, the initial tensile strength of the muscle membrane might be perfectly fine. Dystrophin is there, the DGC complex is there, the muscle can handle the stress, but the cell loses its ability to heal the routine, everyday microtears.
So every time a tiny tear happens, instead of being patched in seconds, it stays open a little too long. A little bit of calcium leaks in, causing a tiny bit of necrosis. And over years, that damage accumulates slowly and relentlessly.
Exactly. This accumulation of unrepaired microdamage leads to a specific clinical presentation, often called a limb-girdle phenotype.
Okay, let's define that. The text mentions the limb-girdle phenotype a few times across several different genetic mutations. What does that actually look like in a patient standing in your clinic?
Limb-girdle muscular dystrophies, whether caused by defects in the sarcoglycans, the repair proteins like dysferlin, or even some of the mild Golgi glycosylation defects, are characterized by a slowly progressive weakness that specifically targets the proximal musculature.
Proximal, meaning closer to the core.
Yes. We call it limb girdle because it affects the muscles around the shoulder girdle, the upper arms, chest and back, and the pelvic girdle, the hips and upper thighs.
But here is the question that always bothers me about these genetic diseases. If the genetic defect, say a mutated dysferlin gene, is present in the DNA of every single muscle cell in the patient's entire body, why those specific muscles? Why do the shoulders and thighs fail while the muscles of the hands, the forearms, and the calves remain relatively strong and functional for years or decades longer?
It is a profound clinical question, and for a long time it was a mystery. Because you're right, the mutated dysferlin or sarcoglycan proteins are absolutely expressed in the distal muscles of the hands and feet as well. Why are they spared?
Exactly.
The prevailing physiological theory is that the pattern of degeneration reflects the pattern of muscle use and mechanical loading. Think about what the proximal muscles, your torso, your quads, your glutes, your paraspinal muscles, actually do all day. They're your primary anti-gravity muscles.
Right, even when I think I'm just standing still, they're constantly working to keep me upright.
Exactly. They are subject to a continuous, chronic, low-level contractile activity just to maintain posture, stabilize your core during walking, and hold your body weight against gravity. This means they are subject to continuous, unrelenting mechanical stress and wear and tear.
And the distal muscles.
Your hands and forearms, on the other hand, are mostly used for intermittent high-dexterity tasks, and then they rest.
Ah, I see. So if you have a baseline defect in membrane stability or membrane repair, the muscles that are under constant chronic load are going to cross the threshold into irreversible damage much, much sooner than a muscle that gets to rest most of the day.
That makes profound logical sense, doesn't it? You wear out the parts of the machine that are constantly running under heavy load first. The anti-gravity muscles simply bear the brunt of the genetic flaw.
It really does. It shows how genetics and physics intersect to create clinical symptoms. Now, this focus on the physical wear and tear of the muscle leads us to a fascinating transition. What happens when the muscle weakness isn't caused by physical tears in the membrane, but by an electrical storm that prevents the muscle from ever relaxing?
Ah, myotonia.
Yes. Let's dive into myotonic dystrophy and the incredibly strange world of toxic RNA. First, let's define the primary symptom here, myotonia. What exactly does a patient with myotonia experience?
Myotonia is defined clinically as severe involuntary muscle stiffness. It's an inability of the muscle to relax promptly and normally after a voluntary contraction. It is not a cramp, and it's not a spasm. The muscle simply remains contracted long after the patient wants it to let go.
The text mentions a classic clinical symptom called the warm-up phenomenon. Paint a picture of what that looks like in a social setting.
The quintessential example is a handshake. A patient with myotonia might reach out to shake your hand. They grip your hand normally, but when the greeting is over and they try to let go, their hand remains clamped shut.
Wow, that must be terrifying at first.
It is. They physically cannot command the flexor muscles in their forearm to relax. They might have to awkwardly pry their fingers open with their other hand, or just wait several highly uncomfortable seconds for the muscle to finally release.
And the eyes, too.
Or, similarly, they might sneeze forcefully, squinting their eyes shut, and then find they cannot open their eyelids again for several seconds.
But it's called a warm-up phenomenon because it gets better with use.
Exactly. If the patient repeatedly opens and closes their hand, shaking it out, so to speak, the stiffness gradually dissipates with each subsequent contraction. After 4 or 5 flexes, the muscle begins to function normally and relaxes smoothly. That is the warm-up effect.
But if they stop...
If they rest for 10 minutes, the stiffness returns in full force on the next movement.
Wait, I need to push back on this for a second. Let's think through the anatomy. If the muscle will not relax, if it's clamped shut, my first thought is that the motor nerve coming from the spinal cord is misfiring. It must be continuously sending a contract signal, flooding the neuromuscular junction with acetylcholine. Is that what's happening? Or is the muscle doing this entirely on its own?
It's a vital distinction, and it was a major question for early neurologists. The stiffness in myotonia is truly myogenic. It originates entirely within the muscle fiber itself. It is completely independent of the nerve supply.
How can we know that for sure? How do you prove it's not the nerve?
You prove it by removing the nerve from the equation. In a clinical or laboratory setting, if you inject a local anesthetic to block the incoming motor nerve, the myotonia persists.
Okay.
Even more definitively, if you block the neuromuscular junction entirely using a powerful paralytic agent like curare, which binds to the acetylcholine receptors and completely stops any nerve signal from reaching the muscle, the myotonia still persists.
So even when the muscle is chemically isolated from the nervous system, it still locks up?
Yes. If you percuss the muscle with a reflex hammer or stimulate the muscle membrane directly with a tiny electrical current, it will go into a sustained tetanic contraction. On an EMG needle inserted into the muscle, after the patient stops trying to contract, you will see spontaneous, loud bursts of repetitive action potentials that wax and wane in frequency and amplitude.
What does that sound like?
It sounds like a dive bomber airplane on the audio monitor. The muscle membrane itself is electrically hyperexcitable. It is firing on its own.
Okay, so the electrical membrane is hyperactive, but the disease is called myotonic dystrophy, so it causes weakness and wasting too. How does this fit into the inherited dystrophies?
Myotonic dystrophy has several very distinctive features that set it completely apart from Duchenne or the limb-girdle types. First, it's an autosomal dominant trait, meaning you only need to inherit one mutated copy of the gene from one parent to develop the full disease.
Unlike Duchenne, which is X-linked recessive.
Exactly. Second, the pattern of weakness is inverted compared to the limb-girdle phenotype. In myotonic dystrophy, the weakness is predominantly distal.
Distal.
It affects the muscles furthest from the center of the body first, the intricate muscles of the hands, the feet, and the lower legs. Patients often present with foot drop or poor grip strength alongside the stiffness.
And reading the text, it sounds incredibly systemic. It's not just a muscle disease.
It's highly systemic, which was another massive clue to its underlying biology. Almost all patients develop early-onset cataracts in their eyes. Affected men commonly experience frontal balding and testicular atrophy.
Wow.
And most dangerously, the smooth muscle and cardiac muscle are involved. They frequently develop severe cardiac conduction system defects, leading to dangerous arrhythmias and heart block.
So we have a single genetic disease causing distal muscle wasting, electrical hyperexcitability, cataracts, baldness, and heart arrhythmias. What kind of genetic mutation can possibly cause such a bizarre, widespread array of symptoms across completely different tissue types? Let's dive into the genetics of this.
The discovery of this mutation represented a massive paradigm shift in genetics. The primary defect in the most common form, myotonic dystrophy type 1, is located on chromosome 19 in a gene called myotonin kinase. But the mutation itself is not a simple deletion of an exon or a misspelled amino acid like we saw in Duchenne or the channelopathies.
What is it then?
It's an expansion of a triplet repeat.
A triplet repeat, meaning a 3-letter sequence of DNA just stuttering over and over.
Yes. In a non-coding region of this gene, the 3' untranslated region, there's a sequence of cytosine thymine guanine, or CTG. In a healthy individual, this CTG triplet stutters or repeats a few dozen times, maybe 5 to 30 times. It's normal structural padding.
But in the disease?
In a patient with myotonic dystrophy, the DNA replication machinery slips, and this segment expands dramatically. The CTG sequence might repeat 50 times, hundreds of times, or in severe congenital cases, thousands of times.
Okay, here's where it gets really interesting to me. It's a non-coding region. It doesn't even code for an amino acid. So how does having extra non-coding stutters in a gene cause the muscle to become electrically hyperexcitable while simultaneously causing cataracts in the eye?
It's a brilliant, fascinating pathogenic mechanism. It represents a completely different way for a gene to cause disease. The disease is not caused by the absence of a required protein like we saw with dystrophin. It's caused by the messenger RNA itself. The RNA is toxic.
Toxic RNA. How does RNA become toxic?
When the cell transcribes this mutated gene, it creates an mRNA transcript with hundreds or thousands of repeating CUG sequences on the end. Now, RNA is a single-stranded molecule and likes to bind to itself.
Right.
These massive, repetitive expanded sections do not behave normally. They fold back on themselves, forming incredibly stable, complex hairpin structures. And because they're so abnormally bulky and tangled, these RNA molecules cannot be exported out of the nucleus to the ribosome.
They get stuck.
They get stuck and they accumulate in the nucleus as dense aggregates or clumps.
So you have these microscopic clumps of mutant tangled RNA trapped in the nucleus. What harm do they do sitting there?
They act like a molecular sponge. The nucleus is filled with specialized regulatory proteins, whose only job is to manage the splicing of other pre-mRNAs. One important family of these proteins is called muscle-blind-like proteins, or MBNL.
Muscle-blind?
Yeah. These toxic RNA clumps happen to have an incredibly high chemical affinity for muscle-blind proteins. They bind to them tightly, sequester them, and soak them up, pulling them away from their normal jobs.
Oh, wow. So the toxic RNA isn't actively destroying anything, it's just kidnapping the cell's splicing machinery.
Exactly.
And because the splicing proteins are all trapped in these clumps, the cell starts missplicing other completely unrelated genes across the entire genome.
Precisely. The toxic RNA ruins the construction of dozens of critical proteins across many different tissues, which perfectly explains the multisystemic symptoms. And in the skeletal muscle, one of the most critical genes that gets severely misspliced due to this kidnapping is the CLC1 chloride channel gene.
The chloride channel.
Yes. Because the splicing factors are missing, the mRNA for the chloride channel is processed incorrectly. The cell produces a defective truncated chloride channel that gets degraded. This leads to a profound, catastrophic loss of functional chloride channels in the skeletal muscle membrane.
And as we'll see in a minute when we talk about channelopathies, chloride channels are the safety blanket that keeps the muscle from firing repeatedly. Without them, the membrane becomes violently hyperexcitable, leading directly to the myotonia. That is an incredibly elegant, if destructive, cascade. The RNA itself is the weapon, causing a secondary ion channel deficiency.
It's a triumph of modern molecular biology that we understand this pathway. We started with a clinical symptom, a patient who cannot let go of a handshake, and over decades traced it down to a triplet expansion, causing a toxic RNA sponge, which alters the splicing of a specific chloride channel. It's breathtaking.
And that mention of the chloride channel is the perfect bridge to our final major topic. We've talked about structural failure in Duchenne and secondary electrical failure from toxic RNA. Now let's look at diseases caused exclusively by primary isolated defects in the ion channels themselves, the channelopathies of skeletal muscle, which encompass the periodic paralyses and myotonia congenita.
Yes. Moving into a realm where the muscle anatomy is perfect, the structural proteins are flawless, but the electrical gateways are compromised at the atomic level.
Before we look at how they break, we need to understand how they work. To really grasp a channelopathy, we have to briefly review the normal, healthy electrical excitability of a muscle fiber. Let's walk through it. What needs to happen electrically for a muscle to contract synchronously?
It's a precisely timed, beautifully orchestrated sequence of ion movements. It starts at the neuromuscular junction. The nerve releases acetylcholine, which binds to receptors on the muscle end plate. This opens ligand-gated channels, allowing a small influx of sodium, which locally depolarizes the membrane.
Meaning the inside of the cell becomes slightly more positive.
Right.
But that local depolarization isn't enough to contract the whole massive fiber.
No. If that local depolarization reaches a critical threshold, it triggers the opening of massive numbers of voltage-gated sodium channels located all along the sarcolemma. These channels swing open, sodium rushes into the cell, and a massive electrical wave. The action potential sweeps longitudinally across the entire surface of the muscle fiber in milliseconds.
But the muscle fiber is thick. It's a 3D cylinder. The electrical signal on the surface has to get deep inside to tell the internal contractile proteins to move. How does it get in?
The cell membrane has these deep, microscopic tunnels that dive radially down into the core of the cell, called transverse tubules, or T-tubules.
Right.
The action potential dives down these T-tubules. As the electrical wave travels down the tunnel, it encounters a specialized sensor, a voltage-gated calcium channel known as the dihydropyridine receptor. This receptor physically detects the change in voltage.
And what does it do when it feels the electricity?
It physically changes shape. And because it is physically coupled to another massive channel on the sarcoplasmic reticulum, the cell's internal calcium storage vault, called the ryanodine receptor, its shape change pulls the ryanodine receptor open.
The vault opens.
The vault opens. Massive amounts of calcium flood out of the storage vault into the main cytoplasm of the muscle cell. That sudden spike in calcium is the final trigger that allows the actin and myosin filaments to grab each other and slide, causing the physical, mechanical contraction.
It's a massive chain reaction. Acetylcholine triggers sodium, sodium triggers the voltage sensor pulls the ryanodine plug, calcium floods out, muscle contracts.
Exactly.
But then, crucially, it has to stop. The muscle must repolarize and relax so you can take the next step.
Yes, relaxation is just as active and critical as contraction. To repolarize, the voltage-gated sodium channels must rapidly and securely snap shut a process called inactivation. Simultaneously, delayed rectifier potassium channels open up, allowing positively charged potassium ions to rush out of the cell, which restores the negative resting membrane potential.
And the chloride.
Critically, this repolarization process is strongly stabilized and augmented by a massive influx of negatively charged chloride ions through the ClC1 chloride channels.
Okay, I have the picture. Sodium starts the fire, potassium puts it out by leaving, and chloride acts as the heavy safety blanket that smothers any remaining embers to keep the cell calm.
That's a great way to put it.
The periodic paralysis and myotonias represent transient episodic derangements in this specific excitability, right? Meaning that between episodes, the muscle works perfectly fine.
That is the defining feature of these primary channelopathies. If you examine the patient between attacks, their muscle bulk is normal, their strength is normal, their reflexes are normal. The derangements are purely episodic.
So what triggers them?
They're triggered by physiological stressors.
Yeah.
Often things like temperature changes, exercise, or most commonly, subtle fluctuations of potassium levels in the blood.
Let's look at the first major category, hyperkalemic periodic paralysis. Hyperkalemic meaning the attacks are triggered by abnormally high potassium levels in the blood.
Yes. For these patients, simply eating a meal with a very high potassium content, like a big serving of potatoes, bananas, or drinking lots of fruit juice, or resting immediately after a heavy bout of exercise, can trigger a devastating attack of weakness.
Why?
The underlying cause is an autosomal, dominantly inherited missense mutation in the gene encoding the main pore-forming subunit of the voltage-gated sodium channel in skeletal muscle.
Let's define a missense mutation. Unlike a deletion where a chunk is missing, a missense mutation means just one single DNA letter is swapped out, resulting in one single amino acid being changed in this massive protein channel. How does swapping one amino acid alter the channel's function so drastically?
It's all about protein folding and mechanics. In hyperkalemic periodic paralysis, that one swapped amino acid creates a subtle inactivation defect.
Inactivation.
Normally, after a sodium channel opens to start the action potential, a small protein hinge swings shut to block the pore from the inside within milliseconds. It must securely inactivate so the cell can repolarize. In this disease, a small fraction of these mutant sodium channels fail to shut completely. The hinge is sticky, or it swings open again inappropriately.
So the door doesn't latch. You have sodium continuously leaking into the cell, keeping the inside slightly more positive, slightly depolarized.
Exactly. And the clinical symptom depends entirely on the severity of that leak. If the inactivation defect is subtle, the slight continuous depolarization makes the cell highly irritable and trigger-happy. It rests closer to the threshold.
So it fires easily.
Yeah, so it fires repetitive action potentials, leading to myotonia muscle stiffness.
But what if it's worse?
If the defect is more pronounced, or if extracellular potassium rises and further depolarizes the cell, the continuous sodium influx causes a massive, chronic, sustained depolarization of the membrane.
And if the membrane is chronically depolarized, it can't reset. The other healthy sodium channels are permanently locked in their inactive state because the voltage never drops low enough to reset them.
Exactly. The cell becomes completely electrically refractory. It cannot fire an action potential no matter how hard the nerve stimulates it. The patient experiences profound weakness or complete flaccid paralysis that can last for hours, leaving them utterly bedridden.
Wow.
Fortunately, during these attacks, the muscles of respiration and swallowing are typically spared, so it is frightening, but rarely life-threatening.
That's hyperkalemic. But the text also details hypokalemic periodic paralysis, which is triggered by low potassium in the blood. And the biophysical mechanism here is absolutely fascinating.
It really is one of the most elegant discoveries in ion channel biophysics of the last few decades. Hypokalemic periodic paralysis is caused by missense mutations in a very specific part of either the calcium channels or the sodium channels in the muscle. Specifically, the mutations occur in the voltage sensor domain.
The voltage sensor domain, that's the specific part of the channel protein that detects the electrical charge across the membrane and tells the central pore when to open or close, right?
Correct. The voltage sensor is a transmembrane segment packed with positively charged amino acids, primarily arginines. Because they are positively charged, when the electrical field across the membrane changes during an action potential, these arginines physically move upward through the lipid membrane.
Like magnets repelling.
Yes. And that physical movement pulls the main pore open. The mutations causing hypokalemic periodic paralysis almost universally involve replacing one of these large, bulky, positively charged arginines with a smaller, neutral amino acid.
What's the physical consequence of swapping a big positive amino acid for a small neutral one inside the membrane?
It literally creates a physical hole. By removing that bulky, charged arginine, the mutation leaves a gap in the structure of the voltage sensor. It creates an anomalous leakage pathway, sometimes called a gating pore current.
So it's a hole.
It creates a tiny microscopic pore right through the voltage sensor domain itself, completely separate and distinct from the main central ion-conducting pore of the channel.
Okay, I have to try an analogy here to make sure I truly understand this anomalous leak because it sounds wild.
Go for it.
Let's think of the entire ion channel as a high-tech bank vault. The central pore, where the sodium normally goes through, is the main massive steel vault door. It works perfectly. It only opens when the manager turns the key.
Okay, I follow the architecture.
The voltage sensor domain is like the thick bundle of security camera wiring that is drilled right through the solid concrete wall next to the vault door.
I like it.
The mutation is like someone pulled out one of the thickest electrical wires from that bundle and replaced it with a very thin piece of string. The main vault door, the central pore, still works perfectly, but now, because the thick wire is gone, there is a tiny physical hole drilled right through the concrete wall where the thick wiring used to be.
Yes.
And water, or in this case, a trickle of sodium or hydrogen ions, can slowly but continuously leak into the vault through that tiny anomalous hole in the wall, bypassing the main door entirely.
That is an exceptional analogy. It perfectly captures the biophysics. The main pore functions normally, but the structural mutation in the sensor creates a parallel leak.
And what does that leak do?
This anomalous leak current allows a slow trickle of positive ions into the cell, which depolarizes the resting muscle fiber. And what is counterintuitive, but central to the disease, is that this specific anomalous leak actually becomes larger and more prominent when extracellular potassium falls.
So a drop in potassium, maybe from eating a heavy, high-carbohydrate meal that causes a spike in insulin, which naturally drives potassium out of the blood and into cells, exacerbates this tiny leak in the wall.
Yes. In a normal person, a drop in extracellular potassium hyperpolarizes the cell, making it slightly more negative and stable. But in these patients, the drop in potassium interacts with the anomalous leak to cause a paradoxical massive depolarization. The resting potential shifts from a healthy negative 90 millivolts to perhaps negative 60 or 50 millivolts.
Yeah.
And just like in the hyperkalemic form, this sustained depolarization locks all the normal sodium channels in an inactivated state, rendering the entire muscle completely unexcitable and paralyzed.
It's astounding. A single atomic change, swapping one arginine, creates a microscopic secondary leak path that can paralyze a human being. The precision, the fragility of that biology is deeply humbling.
It truly is.
Now, there is one more channelopathy you mentioned before we wrap up. Myotonia congenita. We talked about myotonia in the context of myotonic dystrophy and the toxic RNA sponge, but this is a primary channelopathy. It is different.
Yes, myotonia congenita is clinically and genetically distinct. First, the muscle stiffness is present very early, often from birth or infancy, and crucially, it's non-progressive.
Non-progressive, meaning it doesn't get worse.
Right. There is no muscle wasting, no permanent weakness, no structural destruction of the cell, and no other organ involvement like the cataracts or cardiac issues we saw with the toxic RNA.
That's a huge relief.
In fact, these individuals often have muscular hypertrophy. They look very athletic or bodybuilder-like because their muscles are constantly undergoing isometric exercise against themselves due to the myotonia.
Like the famous fainting goats, right, where they stiffen up and fall over.
Exactly like the fainting goats. The text notes this is caused by mutations in the CLC1 chloride channel. This is the exact same channel that was secondarily ruined by the toxic RNA missplicing earlier.
But here, the mutation is a primary defect in the DNA sequence of the CLC1 gene itself.
Yes, the channel is built, but it doesn't conduct chloride properly. The mutation severely decreases the influx of chloride ions.
Right.
Remember, we established that chloride acts as the electrical safety blanket, stabilizing the resting membrane potential after an action potential.
Yes, smothering the embers.
Exactly. Without sufficient chloride conductance to smother the electrical fire, the normal, minor accumulation of potassium that happens in the narrow T-tubules during a single action potential is enough to cause the membrane to depolarize slightly again. This triggers a rapid, uncontrolled burst of repetitive action potentials.
So the initial fire starts normally, but without the heavy chloride blanket to smother it, the sparks just keep reigniting the muscle over and over, resulting in that profound clinical stiffness, the inability to let go of the handshake.
Precisely. It is a pure electrical defect of repolarization.
So if we look back at what we've covered today, we've traversed from whole muscle ischemic starvation to missing structural beams to toxic RNA kidnapping splicing proteins, all the way down to atomic level leaks in ion channels.
You really have.
This incredible synthesis represents the absolute pinnacle of modern neuromuscular science. And that leads us directly into the highlights section of Chapter 57, where the authors pull this massive field together.
The highlights really aim to synthesize the vast landscape we've explored across all three parts of this deep dive on the Enkephalikos channel. We started this journey by defining the motor unit and how diseases are categorized by where the anatomy fails.
Right. We distinguish pure motor neuron diseases like ALS, which relentlessly destroy the nerve cell bodies in the spinal cord, from peripheral neuropathies like Charcot-Marie-Tooth or Guillain-Barré, which attack the long axons or their myelin sheaths, and typically involve both sensory loss and motor weakness distally.
And then today, we clearly separated all those neurogenic disorders from the primary myopathies, where the nerve is perfectly fine, but the muscle fiber itself degenerates relentlessly, often starting in the proximal anti-gravity muscles.
And finally, we looked at the channelopathies, which often present as pure motor weakness or stiffness that can fluctuate wildly over hours, leaving no permanent structural damage between attacks.
Right.
Looking at this entire sweep of pathology, what stands out to you as the most significant scientific takeaway from this chapter?
If we connect this to the broader arc of medical history, what's truly awe-inspiring here is the sheer triumph of molecular genetics. Think about where this field was just a few decades ago. For over a century, neurologists were brilliant observers, but they could only categorize these diseases based on clinical presentation.
Like, this patient is weak distally versus proximally.
Exactly. They have a rash. Their weakness fluctuates with a meal.
It was purely descriptive. They knew what was happening, but they had absolutely no idea why.
Purely descriptive. But starting in the late 1980s and the 1990s, using nothing more than detailed clinical data from affected families and samples of their DNA, scientists were able to discover the specific atomic-level genetic defects causing these syndromes.
It's amazing.
They found the massive dystrophin gene. They mapped the entire DGC structural complex. They figured out that swapping one arginine in a voltage sensor creates a leak.
They completely rewrote the textbook. They turned descriptive clinical syndromes into highly precise molecular mechanisms.
They did. And that discovery created an incredibly powerful feedback loop between basic science and clinical medicine. By identifying the exact genes, we could create small animal models. Mice engineered to have the exact same DMD frame shift mutation or the exact same sodium channel leak.
So we could study them.
Yes. These genetic models have been absolutely invaluable. They allow us to study the microscopic pathogenesis in real time and, crucially, to test entirely new classes of treatments.
Which brings us back to those incredible biological therapies we discussed. Antisense oligonucleotides intentionally manipulating RNA splicing on the fly, adeno-associated viruses acting as microscopic delivery trucks to drop off customized microgenes to replace missing instructions.
These concepts are no longer theoretical science fiction. As the chapter highlights, this deep mechanistic understanding, combined with innovations in viral delivery and RNA technology, has led to transformative successes in human clinical trials. We are witnessing the dawn of a new generation of molecular therapies that can augment, bypass, or silence mutant genes. We are seeing substantial improvements in clinical outcomes for diseases like spinal muscular atrophy and Duchenne dystrophy that just 20 years ago were considered utterly untouchable death sentences.
It really is a profound scientific journey. Reflecting on this three-part Enkephalikos series, we started out in part one by just looking at the simple electrical twitch of a muscle fiber in a textbook diagram. And we ended up today discussing how scientists are actively manipulating viral vectors and designing custom RNA molecules to literally rewrite the genetic destiny of a human cell.
It's the ultimate testament to the power of understanding the mechanism. Once you know exactly how the machine breaks down at the atomic level, you can finally begin to design the precise tools needed to fix it.
I want to sincerely thank you for this absolute masterclass on neuromuscular diseases. Your ability to break down the highly complex biophysics of an ion channel and to help us visualize the massive structural scaffolding of a multi-million base pair gene has been incredibly illuminating.
It has been a true privilege to walk through this complex material with you. The intricacies of the motor unit never cease to amaze me, and I appreciate the deep questions you brought to the table.
And to you, the listener, this officially concludes our deep dive into Chapter 57 of Principles of Neuroscience. Over these 3 sessions, we've covered the motor neurons, the peripheral nerves, the neuromuscular junction, and today, the remarkable muscle fibers themselves. But before we go, I want to leave you with one final thought to mull over.
Always a good idea.
Every time you effortlessly reach out to grab a glass of water, millions of voltage-gated ion channels in your arm are swinging open and snapping shut with atomic precision. The electrical current is diving down the T-tubules, unlatching the calcium vaults.
Millions of times.
Giant, intricate dystrophin proteins are bearing the physical-mechanical load of that movement, distributing the sheer force of the contraction so your cells don't rip themselves apart. And behind the scenes, dysferlin is standing by, ready to rapidly deploy rafts of vesicles to patch the microscopic tears that happen just from the effort of lifting the glass. It's beautiful.
We so often take our movement completely for granted. We only notice our bodies when they fail us. But when you consider the sheer, overwhelming complexity, the absolute biological symphony of the molecular choreography required to successfully contract a single muscle fiber.
It's a miracle.
It is. The real miracle isn't that these highly complex systems occasionally break down in the form of devastating disease. The true staggering miracle is that for the vast majority of us, they work flawlessly, millions of times a day, every single second of our lives, without us ever having to think about it.
Beautifully said.
Next time you go for a walk, take a moment to think about the incredible, microscopic universe working tirelessly beneath your skin just to let you take a step. Until next time, keep diving deep.